Um, Good morning. I am Dr. Kwekube Eduardo. I'll be taking you for general physiology. Um, there are certain books I would recommend that you get them, not all of them, the most appropriate or the one you think it's best you understand. The books I recommend are Review of Medical Physiology by Gannon, Textbook of Medical Physiology by Guyton and Hall, Physiology by Ben et al., Human Physiology by Sherwood, Human Physiology by Vander et al. Now, there are a lot of other physiology books. You can get those ones and read them too. And I believe the most appropriate is the one you understand most. Now, I'll give you the outline of the course. We'll be looking at the levels of organization of the human being. So we'll be looking at the various levels, which are the chemical level, cellular level, tissue level, organ system level, and the organism. Then we'll be looking at body water. Now, we'll look at the total body water, the body fluid compartments, functions of water, and the measurement of body fluid compartments. We'll look at the cellular membranes, the structure of cellular membranes, and we'll look at the membrane transport. And the membrane transport, we'll be looking at diffusion, active transport, vesicular transport, caveolae, osmosis, and filtration. We'll then look at homeostasis, brief overview of that. Since physiology is all homeostasis, we'll look at the brief overview. And under that, we'll look at the homeostatic control systems, components of a control system, and strategies for maintenance of homeostasis. We'll then look at nervous tissue. We'll look at the nerve cells and the glial cells. We'll classify them. And then we'll look at resting membrane potential and action potential. Now, if there is time, we'll look at the muscular tissue. Now, it is my belief that after going through all these, you should be able to describe the levels of organization in the human, describe how the levels are organized, identify the main body fluid compartments and estimate their volumes, describe the structure and components of plasma membranes, describe the mechanisms involved in the transport of substances across plasma membranes, outline the principles of homeostasis and their application in the human, list the main subdivisions of the nervous system, describe the main structure of a neuron, name the glial cells and their functions, describe resting membrane potential and how it is generated, describe how changes in cell ionic movements produce an action potential, and finally, explain propagation of an action potential. Now we're going to start the lecture by defining what physiology is. Physiology is how the body and its parts function. We have the body, it's made up of different parts, and we want to know how these different parts function to keep the human being alive and well. Now we start by looking at the levels of organization in the human. Now this is like putting up a building. You start from your sand, cement, blocks, start putting the structure together till you have a complete building. So we're going to start from the basic level, which is the chemical level. Now, the chemical level is made up of atoms and molecules which come together to form macromolecules. Now, these macromolecules come together to form organelles, and the organelles come together to form a cell. So, from the atoms and chemicals, macromolecules, organelles to the cell will constitute the chemical level of organization, which is the basic level. The next level is the cell or the cellular level. Now, the cell is made up of organelles, and in unicellular organisms, it is the functional unit, the structural and functional unit of the organism. So if you take an amoeba, for instance, it is a cell which has all the components, and it exhibits all the characteristics of a living organism. Now, in multicellular organisms, we have cells coming together to form tissues, which is the next level of organization. So a tissue is made up of cells with similar structures and functions. Now, these will come together to form a tissue. Now, there are different types of tissues. We have epithelial tissues. Now, epithelial tissues will line body surfaces and body cavities. We have connective tissue. Now, the connective tissues would protect certain structures, would hold structures together, and will support structures. Examples of connective tissues are blood, bone, cartilage. Now, these are examples of connective tissues. Then we have muscle tissues. So muscle tissue are the muscles. There are different types of muscle. We have smooth muscle, we have skeletal muscle, and we have cardiac muscle. Now, these muscle functionally, their major function is to aid movement. Now, we are able to move from one part of the room to the other because of the functions of the skeletal muscle. You are able even to write because the skeletal muscles are making your hands and your arms move. So they are basically for functions, for movements, even though they have other functions. Then finally, we have nervous tissue. The nervous tissue are involved in the transmission of impulses. So they are involved in the transmission of information to and from the central nervous system. Now, tissues will come together to perform a function. So if tissues come together to perform a function, would be at the next level of organization, which is the organ level. Now, the organ, as I've said, is made up of tissues performing a particular function. So if you take the heart, if you take the kidney, if you take the lungs, these are all organs, and they are made up of different tissues coming together to perform a function. Now, the next level of organization is the organ system. Now, the organ system is made up of different organs coming together to perform a particular function. Now, if we take the cardiovascular system, for instance, it's made up of the heart. Yeah, it's made up of blood vessels. If we take the reproductive organs, depending on whether you're talking about the male or female, you have the testes, you have the accessory glands. If we talk about the digestive system, it has a lot of organs coming together. And these organs coming together would play a particular function. And this will give us the organ system. And there are about 11 or so organ systems in the human being. You have the integumentary system, we have the muscular, we have the nervous, we have the endocrine, we have the cardiovascular, we have the renal, we have the lymphatic. There are other systems. Now, these systems are interdependent. And when they come together, then we have the organism, which is the human being. So now, recapping, we start with the atoms, molecules, macromolecules, organelles, which constitute the chemical level of the organization. Now, the organelles will come together to form the cell, which is the next level of organization, which is the cellular level of organization. The next level is the tissue level, and the tissues are made up of cells which are similarly structured and coming together to perform a function, a particular function. And I mentioned there are different types of tissues. We have epithelial tissue, the connective tissue, the nervous tissue, and the muscular tissue. Now, different tissues will come together to form an organ. So the tissues come together to perform a particular function. And here we would have an organ. And examples of organs: kidneys, lungs, heart, the GI, the eyes. These are all organs. Then organs will come together to form the organ system. So we have an organ will come together to form an organ system. An example is the digestive system. We have the liver, the stomach, the intestines, the inner, all these will come together to form an organ system. And I mentioned there are about 11 organ systems. Now, these organ systems will then come together to form the organism, and in our case, the human being. Now, these organs are interdependent, meaning that they all work together towards a common goal, which is the survival of the organism and the well-being of the organism. So basically, this is the level of organization in the human. Right. Since it's okay, I'll move on to our next topic, which is body water. Now, if we take an organism, a certain percentage of its weight is made up of water. And the total amount of water in the body is referred to as the total body water. Now, so if we take the human being, a certain percentage of our weight is made up of water. And this percentage of the weight, which is made up of water, is called the total body water. Now, the total body water varies with age, gender, and degree of obesity. Now, age. In infants, the total body water is about 73% of their body weight. Now, there is a gradual decline in the total body water, so that in adults, the total body water is about 60% of the body weight. So here you realize the total body water is higher in infants, and gradually it stabilizes to about 60% in adults. Now, gender. The total body water is higher in males or men compared to women. It's about 55 to 60% of the body weight in men, and 45 to 50% of the body weight in young women. Now, the reason is that young women have more fat than young men, and the adipose tissue would displace water. Right? Fat and water are not good friends. So, as young women have more fat than men, the total body water in men is slightly higher than women. Now, the degree of obesity. The total body water correlates inversely with the body fat. We've seen that in the gender, because there is a higher fat content in females than males, men and women. Total body water is higher in men than in women. So as the fat content in the body increases, the total body water decreases. There is an inverse relationship between fat in the body and the total body water. So if you have two men of, let's say, same age, the total body water in these men will depend on the fat content. Now, if one is obese or one is fatter than the other, then the total body water of the fat individual will be lower than that of his colleague. Now, for fat-free tissues, the total water remains constant, irrespective of the age. So if we take the muscle where we have very little or no fat, then the total body water would remain constant. Now, in individuals or humans, there is a water balance such that at a steady state, water intake should always equal water loss. So the amount of water that we take in will depend on the amount of water that we lose during our activities. Now, the water input will come in the various forms. We get water by taking in water, ingestion, right? Now, Through injection, we have water input. Now, we drink water. So, about 1,500 mils of water is drunk per day. Now, we get water from the food that we eat. We can also get water by the metabolic processes that take place during digestion. Now, during metabolism, one of the end products of metabolism is water. So, these three processes or methods, we do ingest water. That is by drinking, by eating, and by the metabolism of food in the system. Now, the water that we take in depends on the weather. During hot conditions, because we sweat a lot, we lose so much water. So our water intake would increase. Now, if you compare during cold conditions where we don't lose so much water, we take in very little water because we are not losing water. Now, the water intake also depends on our habits and levels of physical activity. Now, if we have a sedentary life where maybe you move from home, maybe jump into a car, get to your school or office, sit in the classroom or air-conditioned room where you hardly sweat or lose water, you don't take much water. But if you are in a field, running around the field, practicing, doing heavy physical activity, maybe your work involves digging trenches, being on the farm, weeding, where you sweat a lot, you'd have to replace the water that is lost through these activities. So water intake would increase. So water intake would depend on the weather as well as our levels of physical activity and habits. Now, how do we lose water? We lose water by sweating. Right. When you are working and the weather is hot, you sweat. And that's one way by which we lose water. Another way by which we lose water is by urinating. And of course, you know, urine is mostly water. Then we lose water through our feces. Now, in addition to these, we have what we call insensible water loss. Now, this water loss is not perceived. We don't see any water through this, but we are losing it. Now, as I speak to you, I'm losing water from my lungs. Right. Because of the heat there, water is lost. Then we are losing water through the skin. Now, this is very different from sweating. In sweating, we see the water. In insensible water loss through the skin, you don't see the water. I mean, I guess that's how come it's called insensible, not perceptive. We don't perceive it. It's like going to hang your wet clothing on a dry line. Give it time, you go back, the clothing is dry. You don't see the water leaving the clothing, but it is being lost. That is not perceived, or you can't perceive this water loss. So we say it is insensible water loss. Now, this is a scale that gives us the water balance we just talked about. Water output each day against water input each day. We have urine, feces, breathing, and sweating. Right. As against water contained in food, water produced by the cells, and water that you drink. So in the human being, there's always a water balance. And this should not be upset so that we maintain homeostasis. Now, we're going to look at the body water, as I mentioned, body fluid compartments. Now, the total body water is not contained in only one compartment. It is distributed between two major compartments. We have the intracellular and extracellular fluid compartments. Now, these two fluid compartments are separated by a plasma membrane or a cell membrane. Now, we're going to look at the various fluid compartments. We have the intracellular fluid. Now, the intracellular fluid is the fluid that is contained in cells in the body. So the cells in the body, so we take the cytoplasm, is intracellular fluid. Now, this comprises 2/3 of the total body water. So we said the total body water is a percentage of the body weight that is made up of water. Now, out of this percentage, 2/3 is intracellular fluid. So let's say if we have an adult whose total body water is 60% of the total body weight. 60%, the 2/3 of 60% will be 40% of the body weight. So the 40% of the body weight will be intracellular fluid. Now, the intracellular fluid is very rich in potassium ions, magnesium ions, phosphate and organic ions, and also rich in protein and ions. But it is low in sodium and chloride ions. Now, the next fluid compartment is the extracellular fluid compartment. So we said that 2/3 of the total body water is intracellular. The remaining 1/3 is extracellular. So this will constitute 20% of the body weight. And is the fluid that is found outside the cells. Now, the extracellular fluid is very rich in sodium, bicarbonate, and chloride ions, and poor in in potassium, magnesium, inorganic ions, and protein and ions. So you realize that the composition of these two fluids, the intracellular and the extracellular, are at variance. When an ion is high in the intracellular fluid, it will be low in the extracellular. When it's high in the extracellular, it's low in the intracellular fluid compartments. Now, the extracellular fluid, it's further divided into 3 subcompartments. We have the interstitial fluid. Now, this fluid surrounds the cells. They are the fluid that immediately surrounds the cells. And this does not circulate. And it comprises about 3/4 of the extracellular fluid. So, we said the extracellular fluid is 1/3 of the total body water. And out of this 1/3 of the total body water, 3/4 of this will constitute the interstitial fluid. Now, the interstitial fluid compartment, or the interstitial fluid, is the only compartment that declines with advancing age. Remember, I mentioned that the total body water depends on age. It's high in the infants, about 73% of the total body weight is water, and this declines to about 60% in an adult. Now, the decline is due to the decline of the interstitial fluid. So it's the only fluid compartment that declines with advancing age. Now, the second sub-compartment of the extracellular fluid compartment is plasma. Plasma is the liquid component of blood, right? And this constitutes about 1/4 of the extracellular fluid. 1/4 of the extracellular fluid. We said 3/4 of the extracellular fluid is interstitial fluid. 1/4 of the remaining quarter is plasma. But I mentioned that there are 3 subcomponents of the extracellular fluid. There is a third fluid compartment, which is not much compared to the plasma and the compared to the plasma and the interstitial fluid. Now, the transcellular fluid is found in epithelial lining spaces. They are not much. And examples of the transcellular fluid are the cerebrospinal fluid, CSF, the GIT fluids, bladder urine, aqueous humor in the eye, and joint fluids. There is a very low turnover of these fluids. So normally their volumes is not considered. In some texts, they are not considered at all. Now, this chart is a summary of what we've just talked about concerning body fluids. Now, if we take an individual, which is about 70 kilograms body weight, about 40, that individual would have about 42 liters of water. So the individual's total body water will be about 42 liters. Why is it 42 liters? We said that the total body water of an adult is about 60% of the total body weight. Now, this individual is 70 kilograms. So 60% of 70 kilograms will be 42 liters. Now, I said that the total body water is divided into 2 fluid compartments: the extracellular fluid, and the intracellular fluid. Now, the extracellular fluid is 1/3 of the total body water. So 1/3 of 42 liters will be 14 liters. The intracellular fluid is 2/3 of the total body water. Now, 2/3 of the total body water will be 28 liters. I mentioned that the extracellular fluid volume. The extracellular fluid volume is divided into three sub-components. We have the interstitial fluid, which is a quarter of the total extracellular fluid. And a quarter of 14 liters is 10.5 liters. We said plasma would be almost, the remaining quarter is plasma. So that will give us almost 3 liters. Then the third compartment, subcompartment, is the transcellular fluid, which is just about 0.5 liters. So this summarizes water, total body water, and the contents of the various fluid compartments. Now there is a question, which is, which of the fluid or compartment makes up edema? Now edema is as a results when the uptake of fluid from the interstitial compartment is less than the filtration from circulation. You know, at the capillary bed, fluid will be moving out from blood into the interstitial space, right? So by the time blood traverses from the arteriolar end to the from the arteriolar end to the venular end, most of the fluid that is filtered from in the capillaries into the interstitium is taken up back into circulation, right? Now the remainder, which is not taken up, will join the lymph, then go back into circulation. Now in situations or under conditions where filtration far exceeds absorption, the water will accumulate in the interstitial tissue, and this will give rise to edema. For instance, if you are traveling a long distance on a bus, you realize that if you take off your shoes, you get to your destination, and at times your feet would find it difficult fitting in your shoes. It means further there's pooling of water, right? It means that filtration would have exceeded absorption, right? And this will cause edema, especially if there is excess fluid in the interstitial tissue, right? When the lymph is blocked as a result of disease, then would have this massive edema, accumulation fluid. So during edema, the fluid moves from the plasma and accumulates in the interstitial fluid, right? Now, in the when conditions are better, when you are moving about, when the skeletal pump is working, right, blood will be moved in the venous system so that there wouldn't be excess filtration. Now, the excess filtration is due to the fact that blood is not pumping as it has to be pumped in the venous system. And this is because when you are in standing or sitting still, the skeletal muscles are not working and they are not pumping up blood. They are not pumping up blood as they should in the venous system. And the venous system being the capacitance system, when it's not moving, Then there would be blood flow at the capillaries will be sluggish, and the filtration in the capillary bed would exceed the reabsorption. So there will be accumulation of fluid in the interstitium, which will result in edema. I hope I've explained or answered your question. Right, I'm glad you've understood. So the fluid that would constitute the edema is in the interstitium. It means that interstitial fluid has increased more than what is supposed to be in that particular region where the edema is. Now, this is the composition of the body fluid compartments. We have plasma, which is extracellular fluid, and intracellular fluid. Realize that sodium is very high in plasma compared to intracellular fluid. Potassium is very low in plasma compared to intracellular fluid. Calcium is higher in plasma compared to extracellular fluid. Magnesium is lower in the intracellular fluid. Chloride is higher in the extracellular, so you can just see the differences. Now we'll get to know why these differences arise and the significance of these differences as the lecture goes on. Now another question has been posted, which of the compartments of the body does osmosis take place? Right. We'll be dealing with osmosis, and we'll talk about osmosis. In the meantime, let's go on with our body fluids. Now, if you take the red blood cell, for instance, red blood cell, it has a membrane, right? And the membrane will allow passage of substances. I'll just answering the osmosis questions. Right. So if you put your red blood cells into a solution that is less concentrated than blood, would have water moving from that solution into the red blood cells. The red blood cell will grow bigger and burst. Right. It will hemolyze. Now that's an example of osmosis. Right. It's an example of osmosis. Now if you put the red blood cell in a solution that is more concentrated than blood, the red blood cell will be losing water through osmosis into the solution, and this will lead to crenulation of the red blood cell. Right. Well, I explain it more when we get to osmosis. I hope in the interim, this has sort of thrown or shed some light on your osmosis questions. So we move on. Now we'll see the functions of water. We cannot function without water. I mean, that's the bottom line. People can decide to fast for a number of days and they will survive. But if people decided to forgo water over this same period, they die. Right. That's how come we have people dying when they are crossing the deserts. Not from food, they die because of lack of water. Right. So without water, the body does not function well and eventually ceases to function. Now these are the functions of water. Transport nutrients and oxygen into cells. So the nutrients that the cells need and the oxygen that the cells need would be carried to them in solution form, and you need water for this to take place. It moisturizes our lungs, the air in our lungs. Now, without that, we'll be breathing in hot air, which would harm our lungs. Then it helps in metabolism. The metabolic activities in the body take place in water. Water protects vital organs in the body. Our brain in the skull are protected from injury by the presence of C cerebrospinal fluid, our spinal cord. The heart would have some film of water around it, the lungs, film of water. So these protect our organs, and water is involved in absorption of water. And it's involved in temperature regulation. When it's so hot, you sweat, and the sweat is evaporated from the body. As the evaporation needs heat, the evaporation will cool the body. So it's involved in regulation of body temperature. Water detoxifies. Right? It detoxifies. Or it will help remove toxins from our body. And water protects and moisturizes our joints. I don't know, maybe you've done some anatomy, you realize that the joints, we have the synovial fluids. Now, these ensure that these protect our joints and moisturize it so that it wouldn't be creaky. And as I've already said, without water, we cannot function. So this chart or diagram summarizes the functions of the water in our body. Next, we want to see how the body fluid compartment volumes are measured. Now, this can be done in the lab, and the measurement is based on the dilution principle. Right, it's based on the dilution principle. Now, assuming you had a beaker containing water of unknown quantity, and you put in a known amount of a certain marker, and you leave this thing to equilibrate so that the marker dissolves in the solution and uniformly. If the final concentration of the solution is determined, we can determine the volume in the beaker. We can determine the concentration. We determine the concentration. We can determine the volume of the water in the beaker by using the dilution principle. Here, the volume will be equal to the amount of solute that is introduced divided by the total concentration of the solution. So an example, if 20 milligrams of glucose is added to an unknown volume of distilled water, and this is left to equilibrate, and the final concentration is 0.05 milligrams per liter, then the volume of the solvent would be equal to the amount of solute that is introduced divided by the concentration. So the volume will be equal to 25 milligrams divided by 0.05 milligrams per ml, which is the concentration. So the volume would be 500 mils. I hope this is understood. I'll go back. We said there is a cup containing an unknown amount of water, and we want to know the amount of water contained in the cup. Right. So let's say we would add a known amount of sugar to the water in the cup. So let's say we add 25 milligrams of sugar to the water in the cup. Now we stir the water and the sugar. We stir till the sugar is dissolved in the water. Then we'll check the concentration of the solution in the cup. I mean, this can be done in the lab. I'm sure you can't do that in your kitchen, but you can do it in the lab. Now, when the concentration has been determined, we want to know the volume of water in the cup. And the volume of water in the cup would be equal to, as you have on your slide now, the volume is equal to amount of solute, which is the amount of sugar we added to the water in the cup. And mind you, we added 25 milligrams of sugar. So the volume will be equal to 25 divided by the concentration, final concentration of the solution. Now when we added the sugar, we stirred it to get a solution. Now we determined the concentration of the solution, and we found that the concentration of the solution is 0.05 milligrams per ml. And the volume of water can be determined using these two parameters, which are amount of solute divided by the concentration of the solution. And we know that we added 25 milligrams of sugar to the water. Now our concentration is 0.05 milligrams per ml. So the volume will be 25 divided by 0.05, and that will be 500 milliliters. 500 milliliters. I can see you've understood this. Right. Now we go on. Now, in some situations, the marker that you use will be metabolized. Right? So meaning that some of it will be lost. So we have to make room for the amount that is lost. This we can do by bringing in a correction factor. Instead of the original formula, which says that volume is equal to amount introduced divided by the concentration. In the next, when there's metabolism, we would have to bring about the amount that we measure, the amount that is lost. So the volume will become amount that is introduced minus the amount that is lost divided by the final concentration. It's about the same formula, but here there is a minor alteration. So let's say after we introduced 25 milligrams sugar, a little of that was lost, and we determined that amount that was lost. So let's say 0.05 milligrams was lost. Now the volume will then becomes 25 milligrams minus 0.05 milligrams divided by the final concentration, which we saw was 0.05 milligrams per ml. So here would have had a correction factor, and we arrive at our volume. I hope this is clear. I hope this is clear. Now, before I'm waiting for your response, but before that, there is a question. Okay, it's clear. Now, there is a question which is, does high sodium and chloride has adverse effect on blood pressure? Yes, it has an effect on blood pressure. You know, sodium movement is always entrained with water movement. So if sodium is being absorbed, water will follow. And when water follows, the blood volume will increase. And that would relate to um, um, that will lead to that would that would lead to high blood pressure. Now, you want me to use examples, right? Examples. Now, these are things that we hardly do in the labs, right? Uh, unless it are in our field of work, right? It is in our field or our line of work. That is when you'll be doing all these things. But we're trying to help you not be doing this, you not be doing this, but we're trying to explain things to you so that you know the importance of these compartments, right? Now, these would be of little value. It's just that it will help you academically when you get to someone mentions it or what that is, then you know it. But as you progress in your physiology, some of these will come up by which and you know how these are done. Now the general physiology bit of physiology is one that would help you explain the systems, right? You remember I mentioned the 11 systems. And these 11 systems, when you are doing these systems, you'll be using the general physiology. So this will help you explain certain phenomena in the body, right? When it is something that you will be seeing on the ward, something that you will need to know, I'll give typical examples. I hope I'm clear on that. Now, there are different markers or dyes that are used for the measurement of the various fluid compartments. Now, these markers, before they qualify to be markers, should have certain characteristics. The markers should be soluble in water. There is no point using a marker that is not soluble in water because we are measuring water, the volume of water. The marker should be measurable. The markers should remain in the compartment being measured. If I'm measuring plasma volume, the marker I introduce should remain in plasma. It shouldn't leave plasma and go into other compartments. It should stay in the compartment that are measuring. Now, the marker should not alter water distribution. It should not affect the distribution of water in the body. The marker should not be toxic. It should not be poisonous. And the marker should not be secreted by the body. If I'm using a marker and the body is introduced or secreting a similar marker, then the result or the outcome of my measurement will be false. Because if I know I introduced X and the body is secreting Y, the amount, the concentration will vary so that the volume I get will not be the right volume. Now, the marker should remain unchanged in the body. And even if it does change in the body, the amount that is changed should be measurable. That is when the correction factor comes in. Right. So basically, these are the characteristics of the markers that are used in the measurement of the body fluid compartments. Now, the markers used will be suitable for certain compartments. So for instance, the total body water can be measured using heavy water. What that means is that you can use heavy water to measure this total body water, but you can't use heavy water to measure plasma or extracellular fluid compartments. And the total body water, we have deuterium oxide or tritiated water. These are heavy water isotopes, right, of hydrogen, different isotopes of hydrogen. So they are heavy, and you can use a scintillation counter to determine the concentration. Now, for extracellular fluid, you can use markers such as inulin, mannitol, or sucrose. It means these cannot be used to measure any other fluid compartments. They can be used to measure only the extracellular fluid compartments. Then plasma volume. You can use any one of these dyes. Radioiodinated human serum albumin, or you can use Evans blue. Now, interstitial volume. Now, this cannot be measured directly. The interstitial volume cannot be measured directly. But we know that the interstitial volume is a component of the extracellular fluid compartment. Now, since we can measure the extracellular fluid compartment and we can measure plasma, we can calculate the interstitial fluid volume by subtracting the plasma volume from the extracellular volume. Because we know that the extracellular fluid volume is equal to interstitial fluid volume and plasma. Since we know extracellular, we know plasma, we can calculate interstitial volume by subtracting plasma from the extracellular volume. Now, the intracellular volume cannot be measured directly also, but it can be calculated by subtracting the extracellular volume from the total body water, because we know that total body water is equal to intracellular fluid plus extracellular fluid volume. So if we measure our total body water, which we can measure using heavy water, and we measure our extracellular fluid volume by using mannitol, sucrose, or inulin, we can calculate the intracellular fluid volume by a simple subtraction. Total body water minus the extracellular fluid volume. Now, we can also determine blood volume. Now, we know blood is made up of both intracellular and extracellular fluid. Intracellular, because blood has cells. We have the red blood cells, we have the white blood cells, we have the platelets. Right. These are all cells. And within these cells, we have fluid. So the fluid in these cells are intracellular fluid. Then we have, of course, plasma, in which these cells are suspended. So blood has both plasma and intracellular fluid. Now, we can measure blood volume by the dilution method. where tagged red blood cells will be injected intravenously and allowed to mix. Now we can determine the concentration. Then we can determine the volume by our method, which is volume is equal to amount introduced divided by the concentration. There is another method which we can use in measuring blood volume as well. Now, this is by the hematocrit method. Now, the hematocrit is the percentage or a fraction of total blood made up of red blood cells. So, assuming we wanted to measure the hematocrit, which we always do when we go to the lab, a blood sample is taken into a tube, and this tube is centrifuged. This results in the separation of the cells from the liquid portion. We put it on a scale, we get to know the hematocrit. Now the total blood can be measured, can be calculated if we measure the plasma volume and divide it by 1 minus the hematocrit. So if our plasma volume is 3 liters and our hematocrit is about 0.4, the total blood volume will be equal to plasma volume, which is 3 divided by 0.6. The 0.6 is what? 1 minus the hematocrit. And this will give us the total blood volume. So this would end our lecture on the body fluids. Any questions or clarifications? So we can proceed. For how long can a human being stay without water? That's a tricky one. Guess it will depend on the individual, but not for long compared to food. Without water for 2-3 days, things will start getting very bad. Things will get very bad. Even for 1 day, it is difficult. We are going to look at cellular membranes. Now, every cell is bothered or limited by a membrane which is called a cell membrane. In animal cells, in plant cells, we have cell membranes. But in addition to the cell membrane, in animals, in plant cells, there is a cell wall. Animal cells do not have cell walls, so it's delimited by a cell membrane. Now, the organelles in the cells also have cell membranes. Now, these membranes are similarly structured, even though there are some variation in the components of the cell. Now, the cell membrane have some functions. Now, it serves as an attachment point for intracellular cytoskeleton. So, in the cell, there are some cytoskeletons. These are tubules. And these tubules attach to the cell membrane to ensure stability of the cell. They are involved in signal transduction. The cell membranes are involved in signal transduction. Now, most physiological activities and biochemical activities in the body are made up of transduction processes, where one information is converted into another information through various biochemical pathways. The cell membrane is the first point for these transduction processes. They are involved in cell-to-cell communication as well. Like information normally will move from one cell to the other. And this information transmission from one cell to the other involves the cell membrane. And they are involved in cell identity. On the cell surface, there are certain proteins which gives identity to these cells. So these proteins the cell membrane would have these proteins. For instance, we know of the typing, blood typing. On the red blood cell membrane are certain proteins which give us a type of blood, so that we are A, O, A, B, or whatever, right? So it's because of the cell membrane which has these proteins, so they are involved in cell identity. Now we'll look at the structure of the cell membrane. The structure of the cell membrane. The cell membrane is a thin structure, it is elastic in structure, and it's pliable. Meaning it's not rigid, it can be molded, turned around, and still will be stable. So it's said to be thin, pliable, elastic in structure. Now it's between 7.5 to 10 nanometers thick. And it's described or said to be a fluid mosaic. It's not a liquid, it's not a solid structure. It is like a thick oil, right? And it's made up of different components. That's why it's said to be a mosaic, right? So it's a fluid mosaic of fluids, of lipids, proteins, and carbohydrates. So if we take the cell membrane, it's made up of lipids, proteins, and carbohydrates. Now, these amalgamate in a fluid form to form the cell membrane. Now, the amount of these components vary from membrane to membrane, and it's important in the role that the membrane plays. We don't have the components being constant in the various cell membranes. They vary, and they also help in the role of the membrane. Now we'll look at the components of the membrane. We said it's made up of lipids, proteins, and carbohydrates. First, the lipids. There are three major classes of lipid molecules in the cell membrane. We have the phospholipids, we have cholesterol, and we have the lipoproteins. Sorry, glycolipids. Now, these occur in different ratios, right? The membranes have these lipids in different ratios. First, we'll look at the phospholipids. There are four major phospholipids present in the cell membrane. Then there is one that occurs in very minute quantities. We have the phosphatidyl serine We have phosphatidyl choline, we have phosphatidyl ethanol amine, and we have sphingomyelin. These are the major phospholipids. We have phosphatidyl choline, phosphatidyl ethanol amine, phosphatidyl serine, and we have sphingomyelin. In addition to these phospholipids, there is one that occurs in very little quantities, but very significant in cellular transduction. These are the phosphatidyl inositols. They are also present in the lipid bilayer. Now, the structure. Each phospholipid has a head. This is the head and two tails. The head is made up of phosphates and the tails made up of fatty acids. The head is hydrophilic, meaning it's water-loving. The tail, the fatty acid tails, are hydrophobic. Now, because of this structure, the phospholipids are arranged such that the hydrophobic tails are sandwiched between the hydrophilic heads. So they will form a lipid bilayer. Now this is a layer of phospholipid. That's a layer of phospholipid. Because the tails are hydrophobic, meaning they hate water, they are sandwiched between the hydrophilic heads, water-loving heads. So this is the head, this is another head. Now the heads are exposed to the cytoplasm of the cell, which is water, and the extracellular fluid, which is water. So the heads will face there, and the tails, which hate water, will be hidden from the water components in the cytoplasm and the extracellular fluid. So we see the hydrophobic tails sandwiched between the hydrophilic heads. So we have a lipid bilayer, phospholipid bilayer. This lipid bilayer is, as I mentioned, fluid in nature. It's fluid in nature. It's not a rigid structure. So the layer is held together by non-covalent bonds between the tails, hydrophobic tails. Now, because it's a fluid, there are lateral movements in the lipid bilayer. You have the phospholipids moving in this direction or that direction. Lateral movement. I mean, this type of movement is frequent. And in rare cases, we have what we call the flip-flop movement, where a phospholipid in one layer would move to the other layer. For instance, this will move up there and that will move out down here. So we have a flip-flop movement. Now, this type of movement is rare in the lipid bilayer, meaning it hardly occurs. Now, this lateral movement is very frequent. It occurs. Now, if this layer is breached, these will flow and come together, right? So, basically, that's the phospholipid bilayer. Now, the other lipid component is cholesterol. The cholesterol also has a water-loving portion and a water-hating portion. And they are found integrated in the lipid bilayer. They interdigitate with the phospholipid molecules. So that's cholesterol, and these are phospholipids. They are found in between the phospholipid molecules. Now, cholesterol would decrease the fluidity of the phospholipid bilayer if it's present, even though the phospholipid is fluid. The fluidity is reduced, and it gives stability to the phospholipid bilayer. Cholesterol would give stability to the phospholipid bilayer and would decrease the flow of polar molecules across the lipid bilayer. If it's present, that polar substances do not easily flow across the phospholipid bilayer. The cholesterol is very important in the maintenance of the lipid bilayer. It ensures that the temperature to which the temperature at which the lipid bilayer would solidify is depressed. Meaning, if cholesterol is present, the fluid nature of the lipid bilayer would be enhanced. Because at certain temperatures, fat or lipid solidifies. Presence of cholesterol within the lipid bilayer prevents the solidification of the lipid bilayer, even at very low temperatures. Now, the third component of the the lipid present is the glycolipids are interaction between carbohydrates and the phospholipids salts. Now the proteins. There are two major classes of proteins in the lipid bilayer. We have the integral proteins and the peripheral proteins. The integral proteins are attached to the hydrophobic regions of the lipid bilayer. For instance, this is an integral protein. This is an integral protein. That's an integral protein. That's an integral protein. They are embedded in the lipid bilayer. Now, some integral proteins are transmembrane proteins, meaning they traverse the whole of the lipid bilayer. So that's a transmembrane protein. That's a transmembrane protein. But this is not the transmembrane protein. It is just embedded in the lipid bilayer. But this will cross the lipid bilayer. So they are said to be transmembrane proteins. The proteins have also water-loving regions and water-hating regions. And the water-hating or hydrophobic regions would integrate with the hydrophobic regions of the lipid bilayer. So please notice the difference between these types of integral proteins. One is just stuck in the lipid bilayer. The other will cross the lipid bilayer. Those that cross the lipid bilayer constitute what we call the transmembrane proteins. Now, all these are integral proteins because they interact with the lipid bilayer by attaching to the phospholipid region, the hydrophobic region of the lipid bilayer. Then we have the peripheral proteins. The peripheral proteins do not interact with the hydrophobic regions of the lipid bilayer. They interact with the hydrophilic ends of the integral proteins, or they would interact with the hydrophilic ends of the phospholipid bilayer. For instance, if we look at this is a peripheral protein, and it's interacting with an integral protein. It is not attached to the hydrophobic region. That's a peripheral protein. It interacts with just the hydrophilic heads of the phospholipid bilayer. Now, because these peripheral proteins do not or are not integrated into the lipid bilayer, the hydrophobic ends of the lipid bilayer, they are easily dislodged by alkaline solutions or by concentrated salt solutions. Now, the proteins that are found in the lipid bilayer have a lot of functions. And these functions include channels. They may serve as channels. They may serve as carrier proteins. They may serve as receptors. They may serve as enzymes. They may serve as cell adhesion molecules. They may serve as antigens. They are involved in cell surface recognition, and they form regions where the cytoskeleton can attach. Now we have the carbohydrates. We said the lipid bed, the plasma membrane is a fluid mosaic of lipids, proteins, and carbohydrates. The carbohydrate composition in the lipid bilayer is low, about 3%. And the carbohydrates that are found in the lipid bilayer are short-chain carbohydrates. Right. Now, the carbohydrates would interact with the phospholipids to form glycolipids, or they would interact with the proteins to form glycoproteins. So here we have a carbohydrate here interacting with the phospholipids to form glycolipids, or interacting with proteins to form glycoproteins. So, in effect, would have a lipid bilayer made up of the phospholipids, and in between the phospholipids we have our cholesterol, and we have our proteins, then we have our carbohydrates. Now, the carbohydrates that are found in the cell membrane would constitute a coating on the surface of the cell membrane. They form a coat on the surface of the cell membrane, like this. Now, this carbohydrate coating on the surface of the cell membrane is called glycocalyx. Now, the carbohydrates, as you've seen, are not found in the cytoplasmic end of the lipid bilayer. They are found at the extracellular end of the lipid bilayer. So we have a carbohydrate here, which will form a coat on the surface of the plasma membrane. And this coat of carbohydrate is called glycocalyx. Now, the lipid bilayer is said to be asymmetrical, meaning if we split the two layers, it's a bilayer. If we split the two layers, we wouldn't have and one layer being the mirror image of the other. One layer may contain certain molecules, structures that will be absent in the opposite layer. Now, I'm trying to show you something. Assuming my hands constitute a lipid bilayer, that's the cytoplasmic end, that's the extracellular end, our lipid bilayer. If I split it, this one wouldn't be a mirror image of this one. It means there are structures that will be present in this that are not present in this. So they are said to be asymmetrical. Now, using my hand, for instance, there is a ring present here, it is absent here, so it's asymmetrical. Now, if we take the lipid bilayer, on the apical surface, there may be certain structures present that were not present at the basal surface. On the lateral side, there may be certain structures present that are not present on the other side. So they are said, the plasma membrane, in addition to being thin, pliable, and fluid in nature, is said to be asymmetrical as well. So this brings us to the end of our plasma membrane or cell membrane.